Dear all, welcome to this video. Please hit the subscribe button so that you automatically get updates for upcoming videos and lectures. Thank you. Enjoy watching.
So now we will discuss the classification of bacteria. The bacteria can be classified on the basis of following criteria, that is shape, cell wall, flagella, nutrition, temperature response. Now first we will see the shape. On the basis of shape, bacteria are classified into four types. Four important types are Coccus, Bacillus, Vibrio and Spirilla. Other than that, the spirochetes, actinomycetes, mycoplasmas are also some classes which comes under other classes, but four major are Coccus, Bacillus, Vibrio and Spirilla. We will see each one of them. Coccus stands for circular or archeon, that has spherical, oval or round shape. So those bacteria which are round in shape, those come under the class Coccus. Coccus is singular, cocci stands for plural. And we will see according to the different arrangements, these cocci are again classified, sub-classified into uh, types like diplococci, where cocci are present in groups, that is in pairs. Example is Streptococcus pneumoniae. And Staphylococci, these are grape-like structures of cocci. Means not in pair but more than two. That's the example is Staphylococcus aureus. Tetrads is a group of cocci, four cocci together arranged in same plane, only singular plane. The example is Micrococcus species. Sarcina is what? It is again group of bacteria or group of cocci together, that is 8 cocci together and in a cube-like arrangement. Means more than one plane it is present, so that is Sarcina. And example is Sarcina ventriculi. Then Streptococci is chains of cocci, singular chains. An example is Streptococcus pyogenes. So here you can see this is a singular coccus. Diplococcus means in pairs. Staphylococcus means grape-like structure in many but in one plane. Streptococci means chain of cocci. Sarcina means 8 cocci in two planes. And tetrad is in singular plane 4 cocci. Next is Bacillus. Bacillus is a gram-positive rod-shaped bacteria. We'll be seeing what is gram-positive. And these are usually obligate, aerobe or facultative anaerobe. Now, what is the difference between the word obligate and facultative? Obligate is compulsory and facultative is optional. So, they are compulsorily aerobe, that is, they require oxygen for their growth. But in absence of oxygen, they can also grow. So, that's why it is called as facultative anaerobe. Now, these are the arrangements of Bacillus. Again, Coccobacillus is what? Usually, Bacillus stands for rod shape. Okay, if any bacilla which is rod in shape, that's why it is called as Bacillus. Coccobacillus is rod in oval shape. That is somewhat spherical but somewhat oval. So that's why it is called as Coccobacillus. Bacillus here you can see it is complete rod shape. Two bacillus together with this one in pair, that is Diplobacillus. Palisades means clusters of bacillus, and Streptobacillus stands for the chain. So there are some terms which are popular. Diplo means two, Strepto means chain. So that these prefixes remain common in all the cases. Only the suffix change according to the shape. So bacillus stands for rod. So that is why the suffix will change. If it is circular, it will form cocci. Next class is Vibrio. Vibrio is gram-negative bacteria. Vibrio stands for comma, rod-shaped comma. So now here you can see these are rod-shaped and they have a flagella. Okay, this is a rod shape but in comma shape, not a straight rod. Are you getting? So that's why it is called as Vibrio, and all members are motile and have flagella. Next class is Spirilla. Spirilla stands for spring-like structure. So here you can see these are spring-like structures. That's why it is called as helical shape bacteria. Again, in Spirilla, there are two types, Spirillum and Spirochete. Spirillum example is Campylobacter jejuni and Spirochete is Treponema pallidum, which causes syphilis. Now, on the basis of cell wall, again the gram-positive bacteria, there are two types of bacteria, gram-positive and gram-negative. So, this differentiation has been made depending upon which bacteria can retain the Gram stain. So Gram was a scientist who prepared a stain where first you use crystal violet stain, and then you apply alcohol to it, then you apply iodine solution, wash it off, and finally again you apply saffron into it. So all those bacteria which appear violet in color are called as Gram positive bacteria because they retain the initial stain, that is the primary stain, and all other bacteria which do not retain the stain and they appear pink, because they retain the saponin stain, those are known as Gram negative bacteria. Now, this differentiation occurs due to the cell wall composition. So, we will see first of all what is present in Gram positive bacteria. Now, Gram positive bacteria are made up of layers of peptidoglycans which are thick and rigid. Now, what is this peptidoglycan made up of? It contains diaminopropylic acid, which is also called as DAP, uramic acid, and teichoic acid together. So three acids, diaminopimalic acid, uramic acid, and teichoic acid together form DAP. Together form peptidoglycan. Then again this peptidoglycan is divided into three parts. First of all, it has an alternating N-acetyl glucosamine and N-acetyl uramic acid backbone. Then this backbone is again attached to a tetrapeptide side chain of N-acetyluramic acid, and again there is a pentapeptide cross bridges in between them. So this structure whole forms the peptidoglycan. Now in case of gram-negative bacteria, as I've already discussed, they appear pink in color, due to the stain saffronin under microscope. Now these gram-negative bacteria, they possess peptidoglycans, but very few layers of peptidoglycans, and they are majorly made up of lipopolysaccharides. Okay, that is lipoproteins and phospholipids. The peptidoglycans remain bound to the lipoprotein of the membrane. Means these peptidoglycans, there is combination of both lipopolysaccharides and peptidoglycans. So peptidoglycans, but they are present in small portion. There are channels made up called as porions. Okay, which has been made from pores. So these channels, they help in permeability of food, nutrition, water, vitamin, B12, and iron. So these channels are present only in gram-negative bacteria. They are absent in gram-positive bacteria. So here is a difference in structure of gram-positive, gram-negative. Here you can see in gram-positive, the peptidoglycan layer is larger one, and cell membrane is smaller. It is complete peptidoglycan. Whereas in gram-negative bacteria, here you can see that there is a peptidoglycan small layer, then there is a lipoprotein layer, and finally that is a lipopolysaccharide layer. And these are the surface proteins. Now, number of layers. So in gram-positive, there is only one layer. In gram-negative, two layers. Thickness. Obviously, gram-positive is thick, 20 to 80 nanometer. Yet is thin, 8 to 10 nanometer. Outer membrane is absent in gram-positive, but gram-negative contains the lipopolysaccharide outer membrane. Periplasmic space is present in gram-positive in some, but in all gram-negative it is present. Chemical composition, obviously peptidoglycan, teichoic acid, and lipoprotein acid. Here it is only lipopolysaccharide, lipoprotein, and peptidoglycan. Porine channels are absent in gram-positive, they are present in gram-negative. Lipids are less in gram-positive, more in gram-negative. Peptidoglycan is more in gram-positive, less in gram-negative. Permeability of molecules is more in gram-positive, less in gram-negative. And resistance to molecules is less in gram-positive and more in gram-negative. On the basis of nutrition, the bacteria are first of all classified into autotrophic and heterotrophic. I guess everyone knows the difference. Autotrophic is the bacteria which can synthesize their own food in the presence of CO2 and water and light, obviously, because they produce the food in daytime and they have a green pigment in them. Whereas heterotrophic bacteria, the food is obtained from this autotroph, so they don't require the green pigment, and they will get the food available all the time. Okay. Now, under this heterotrophic again, there is subclasses, that is holozoic nutrition, saprophytism, and parasitism. Holozoic nutrition, which means the organisms which will feed on ingested solid organic material, and then it is digested into their bodies. Okay. Then saprophytism are the almost normal food, whatever they will take, like directly plants and all. So those will be holozoic. Saprophytism means These are the saprophytes which feed on dead and decaying matter. They don't feed on living matter. And they include the bacteria and fungi which digest the food externally before the nutrients are absorbed. Okay, nutrition absorption takes place later and externally digestion takes place. Here the digestion takes place internally in holozoic nutrition. And in parasitism, it obtains the nutrients from living organisms, and the parasite obtains the nutrients by living on or in the body of the host. Example is fleas, lice, and tapeworms. Okay. And there is one more class which I haven't included this slide, but there is one more class depending upon the temperature. So the bacteria can be classified into three classes: psychrophiles, mesophiles, and thermophiles. Psychrophiles are the bacteria which can live in lower temperatures, that is up to minus 15 degrees Celsius, 0 to 15 degrees Celsius. Then mesophiles are the bacteria which can live up to 25 degrees Celsius, and thermophiles are the bacteria which can live at a temperature of around 70 to 80 degrees Celsius. Actually 70 to 80 is a hypothetically it is said, but usually up to 60-65 degrees Celsius these bacteria can be sustained. So those are known as thermophiles. Thank you. So here we have completed the classification of